if you're that guy in biology who's like, dude, what's the difference between transcription and replication? Because we both have, I mean, I don't get this stuff. We have RNA polymers, we have DNA polymers. They're both making these polynucleotides. I don't understand what's happening. Well, by the end of this video, I hope you're not saying the same thing. So we're going to talk about the differences between transcription and replication. We're going to start this video off by discussing the end goals of transcription and replication first. So replication is really just a process where we have linear DNA. So this is a nucleosome. A nucleosome is when we have 8 histone molecules, and then we have double-stranded DNA wrapped around it. That's going to be unbinded, and then it's going to allow DNA to replicate itself. It's going to cause DNA is semi-conserved, which means that each one of these strands can be used as a template to form its complementary base pairs. And because they're complementary, they're going to be the same thing. And this is another representation through color. We have two strands. Each one of them, so this strand is used over here, and this strand is used over here, can be used as a template to produce a daughter DNA strand, which is going to be both complementary to one another. So this is the end goal of replication, just to produce 2 daughter cells, or I'm sorry, daughter DNA, and they both are identical to one another. This happens due to when cells are replicating. So transcription's end goal is really to produce DNA, which is coding RNA. Coding RNA is one example, and probably and the only example for in terms of translation reasons is mRNA. So mRNA is the end goal for DNA, for transcription. And then we also talked about non-coding RNAs and coding RNAs, and we have discussed that RNAs can also produce non-coding RNAs, which are functional RNAs, but they don't have anything to do with translation or the synthesis of proteins. And an example of non-coding proteins would be tRNA or ribosomal RNA, and you can look that up on my channel. I have a video for that. So let's get on to the similarities and differences. The biggest similarity between these two is that they both are template-based polymerization. What I mean by that is it's polynucleotides are being formed, and it's the hydrolysis of pyruvates, and it's a forming of the nucleic backbone. We talked about that before. The nucleic backbone is the sugar and the the orthophosphate that's attached to it, and these two will remain the same throughout the entire DNA. And we also talked about how 2 nucleotides will bond to one another through phosphodiester bonds, and these phosphodiester bonds are, and this is a very brief overview, is caused by the nucleophilic attack through the third carbon or the hydroxide on third carbon is going to give off its electron to the phosphate. There's a nucleophilic attack, and it's going to help form this structure, and the pyrophosphate is a leaving group. We have this phosphodiester bond over here. So if you're trying to understand how this exactly works, search it up on my channel. Just search polynucleotide formation, and you'll understand how this works. But for the function or the purpose of this video, this is just a reference to how the mRNA is being synthesized for the purpose of transcription, and then how the complementary new strand is being formed for replication. The difference between the two is that, which is going to help understand if a DNA polymer is needed or an RNA polymer is needed. So DNA replication needs an RNA primer because it's using a DNA polymerase. Excuse me. So a DNA polymerase requires a RNA primer. Without it, it cannot synthesize the new complementary strand. Whereas transcription needs a RNA polymerase, and the RNA polymerase does not need the RNA primer. And that's a really good way to distinguish what is needed for what. Another similarity, which I just want to scream out to you, is both synthesized from the 5' to the 3' direction. I hope you remember that, because this is very, very important. 5' to 3' is the direction of synthesis for pretty much every time you have polynucleotides being formed, and that should be obvious, and that should be obvious by now. I'm not gonna. The fourth difference is that RNA polymers lack exonuclease activity like the DNA polymer has. And this just means that transcription is going to have more mistakes and more mutations than DNA polymerase. And the reason being because DNA polymerase has exonuclease activity. Exonuclease activity refers to the ability to proofread the DNA. So it's what really happens is so 5' to 3' synthesization happens, and then the DNA polymer has the ability to go back once again and check everything is fine, and that's an overview of what I just have explained too many times before. So I recommend you go search my DNA polymerase videos, and you'll see what I'm saying. And then there are two kinds of viruses that are, so here's a question to ask. Okay, here's a question for you to ask. The two kinds of viruses are found in a sample. So there's virus A that relies on DNA polymerase, and virus B relies on RNA polymerase, which is expected to have a higher mutation rate. Well, the answer to this question really just lies in the realization that DNA polymerase has the ability of exonuclease activity, and RNA polymerase does not have the ability of exonuclease, which is proofreading. So, therefore, you can understand that virus B is going to have a higher rate of mutation. This is what these viruses love to do. So virus A will have a less mutation because it has DNA polymerase, which has exonuclease activity to 3' and to the 5' generally. Exception is polymerase 1. Oh yeah, exactly. So exception to this is DNA polymerase 1, which has the ability to have proofreading in both directions, the 5' to 3' and the 3' to the 5' end. Another similarity between these both is both have codons, which signal start of replication. And this is okay. So these both have not start codons exactly, but these both have codons which will signal start of replication or the transcription. So transcription has a start site, and these start sites have nucleotides which are called promoters, whereas replication has the start point of replication is called the point of origin, and of course RNA primers are used for both templates of strands that are needed to replicate.